Medica Corporation is pleased to highlight the Easycell Assistant, an affordable way to simplify manual differentials. When the carousel is complete, the technologist selects the Review option. This results in a display of a file for each of the slides run on Easycell. The technologist selects a slide file to review. The Easycell located 7 different types of white cells for this patient. They are listed in the category buttons on the left side of the display. The technologist examines the segmented neutrophils and determines that they are all classified correctly. The tech identifies toxic granulation in the segmented cells. The technologist next verifies bands and switches to medium-size images. This is followed by a quick inspection of lymphs. Monos, EOs, basophils, variant lymphs, and nucleated red blood cells. The technologist next examines non-cell categories, starting with smudges. Unknowns are viewed in the small image size. Examples of unknowns are broken cells, platelets, and stain. Easycell has placed several immature cells in the Other category. The technologist reclassifies the cells and moves them to the correct category. To simplify this task, images can be moved as a group instead of moving one at a time. These are metas. These are Milos. These are blasts. The technologist returns to the blast category and adds one image to the report. In the same way, one Milo is added to the report. The technologist moves on to the red blood cell screens to verify morphology and the platelet estimate. The technologist is required to examine all red blood cell images within 4 screens. On this screen, the technologist observes several morphological anomalies and records the information in the columns to the left. The size of one red blood cell is measured by clicking on one edge and dragging to the opposing side. This cell is identified as a macrocyte. Other size and shape features are recorded in the morphology columns on the left, such as hypochromatic, polychromatic, microcytes, schistocyte. And on the second page, a giant platelet is found. Finally, the technologist records an increased platelet estimate and the presence of a giant platelet. Next, the technologist adds a custom comment to the report, noting that pear-shaped cells were observed. The technologist opens the report to confirm that images and morphologies are recorded. It is recommended that quality control, or QC, be run once per day depending upon lab policy. The QC summary of findings shows the number of cells found, cells found to be true positive and true negative, and false positive and false negatives. The technologist enlarges images for closer examination. True positives are inside green circles. Non-cells are inside black circles. The technologist has examined all the QC screens and made adjustments where necessary. The QC percentages are updated to reflect these changes. The technologist saves the review. Once saved, Easycell Assistant returns to the list of Easycell slide files. The Easycell Assistant has a number of key features. It presents pre-classified digital images of white cells, red cells, and platelets, saving time. Easycell has a 30-slide carousel for walk-away operation. In addition, a stat slide position provides quick turnaround for critical samples. Easycell reads sample barcodes. Alternatively, a snapshot is taken of non-coded labels. Easycell Remote enables multiple pathologists to review results from their offices. For further information about Medica's Easycell Assistant, contact the following: